Good evening. This is Ion Channel and Neuronal Communication. My name is Hannah Antosh. This presentation is for Dr. Karma Cook for Chemistry 530, Concepts of Biochemistry. Before we begin the bulk of our presentation, I wanted to take a minute and introduce you to the purpose of this lesson. There are 6 main points that we will be talking about today. The first one is neuronal communication. The second point is to identify the function of channel proteins within ion channels. Third, to identify the ions that affect neurons, specifically sodium. 4, to explore the structure of a neuron. 5, explore the way that ions affect neuronal communication. And finally, a few real-world applications. So what is neuronal communication? Neuronal communication is when neurons utilize the existing membrane potential that occurs with an uneven distribution of sodium and potassium ions in the intracellular fluid and the extracellular fluid. When there is an uneven distribution of these ions, this is called a membrane potential. Membrane potential is typically maintained at a pretty constant resting level. However, when an action potential is required, permeability changes can occur, which creates higher permeability levels for potassium and sodium. There are things called voltage-gated channels. There's 2 specific ones. Only one that we're really going to talk about today, though. The first one is a voltage-gated sodium channel. This is going to be our main focus. The second is a voltage-gated potassium channels. Both of these channels are very sensitive in changes to the voltage. These voltage changes signal when it is time to open and close those voltage-gated channels. Channel proteins. Channel proteins form hydrophilic pores that transverse the membrane, leading from one side of the membrane to the other. There are several different types of channel proteins, such as gap junctions, porins, and ion channels. Of these 3, we will spend the most time today talking about ion channels. That's the main focus of this presentation. Ion channels are specifically concerned with the transportation of inorganic ion movement, meaning the movement of ions into the cell from the extracellular matrix. These ion channels are highly selective. They only allow that, especially in the case of sodium, voltage-gated sodium channels, they only allow that specific ion into the cell. They are narrow, and they are capable of opening and closing when necessary. If we think back to maybe a freshman biology class, you probably remember the talk about concentration gradients. You probably talked about it when you learned about osmosis or diffusion. Remember that with concentration gradients, ions, in concentration gradients, things tend to flow from high levels of concentration to low levels of concentration. This is called passive transportation because it requires no energy. Ion channels can only use passive transportation because ion channels are unable to be paired with an energy source that is required to do active transport. So this means that sodium and potassium can only flow from through ion channels from areas of high concentration to areas of low concentration. And this becomes very important when we talk about the neurons. There are several ions that are involved in neural communication. These ions include sodium, potassium, calcium, chlorine, and chlorine. Neurons specifically use ion channels to communicate. To talk about ions and neurons, we need to talk about the neuron structure. This graphic is a great way to show that neurons are highly complex cells, and they don't look like normal animal cells. If you've ever been in a biology class, you've probably seen a poster of an animal cell. They are typically going to be kind of a round shape with lots of organelles inside. Neurons don't look like that. These are highly specialized cells that are typically found in the central nervous system. Their significant structures include their dendrites, which are these little antler things over here, antler-looking things, I should say, over here. The dendrite is where neurons take in chemical input from other neurons. Neurons also have a cell surface membrane, and within this cell surface membrane, we can see the voltage-gated sodium channel. This voltage-gated sodium channel allows sodium ions to enter into the neuron, and this begins an excitatory action potential that leads all the way down to the terminal, down here. This voltage-gated sodium channel allows these sodium ions in, and then, which again, as I said, begins the excitatory action potential, which stimulates the required neurotransmitter to be released all the way down here at the axon terminal. The axon terminal is the part of the neuron that is used to communicate with other neurons via neurotransmitters. The surface membrane that I just talked about of neurons allows ions to move through the membrane from one side to the other by that voltage-gated protein channel. This process allows the neuron to experience, an excitatory action potential, which again triggers that neurotransmitter. This is the most expedient form of intracellular communication. Typically, neurons are completely separate from one another. They are not connected. They typically will send neurotransmitters across something called a synaptic cleft, which we'll talk about in 1 minute. Because they are typically separate from one another, they are not able to directly share their electrical or chemical signals, except for electrical synapses. This is where ion-conducting pores can actually join the intracellular compartments of nearby neurons. Most of the time, when you hear a rule in science, you can pretty much bet that there's at least 1 or 2 exceptions. When I teach freshmen, anytime I teach them a new rule, I always warn them, be aware for at least 1 to 2 exceptions. But this electrical synapse allows for a direct ion flow from neuron to neuron without having to deal with that synaptic cleft. Now, electrical synapses are not typically a commonplace occurrence, at least according to Levenger. So they're not super something that we need to spend a lot of time on today. So ions flow because, the flow of ions, again, allows the release of a neurotransmitter. And this neurotransmitter allows for communication between cells. Think of, if you are a word picture person, think of this like 2 teenagers in a class where they're not supposed to be talking. Right? Teenager 1 over here just really wants to talk to teenager 2. Teenager 1 isn't allowed to talk to them directly, right? They're not touching. They can't communicate directly. So what does teenager 1 do? Teenager 1 gets a vesicle and fills it full of neurotransmitters. Now, the vesicle acts like a piece of paper. Neurotransmitters act like words on a paper. So teenager 1, aka the presynaptic neuron, sends this little message. Maybe the message is a note about a football game, or if it's 2 love-struck teenagers, maybe it's a love note. Who knows? But they send their message across this cleft, and these messages meet on the other side, and they go to the postsynaptic neuron. Again, as I said, these synapses are not connected. They are within 20 to 50 nanometers of each other. And the neuron that is sending the neurotransmitter is called the presynaptic neuron. This is the releasing transmitter. The one that is receiving is called the postsynaptic neuron. Again, neurotransmitters release via the vesicles across the synapse to the presynaptic neuron. From the presynaptic to the postsynaptic. That neurotransmitter, on its journey, it's encased within the vesicle. It travels, or traverses is a much more dramatic way to phrase it, traverses this synaptic cleft and lands in and binds to the ligand-gated ion channels and the G-coupled protein receptors. The gene-coupled protein receptors that are located on the presynaptic neurons are able to change the function of the voltage-gated ion channel, as seen by this diagram down here. We can see that this G protein coupled receptor is then able to modify this voltage-gated sodium channel, telling it to open or to close depending on what the neuron needs. Because this G protein coupled receptor is able to influence the voltage-gated sodium channel, this GPCR is then able to actually moderate the flow of neurotransmitters to either increase or decrease the flow. There are, of course, several real-world applications of this concept. The first one to talk about today is schizophrenia. Schizophrenia is one that is quite personal to me. One of my grandmothers suffered her whole life on and off from bouts of schizophrenia. Schizophrenia is a mental disorder that has both environmental and genetic factors in its formation. It is a psychotic disorder, so patients typically will experience psychosis. Clinical signs of schizophrenia tend to manifest between 16 and 30 years. 30 years being on the older end of it, definitely. It tends to manifest quite a bit earlier than 30. I think my grandmother manifested in her late teens, early 20s, somewhere in there, which is pretty typical. The symptoms of schizophrenia are quite broad. They can include the general hallucinations that are characterized in popular culture. They can also include delusions, paranoia, as well as cognitive and social defects, among other things. Researchers tend to think that the hyperactive state of the dopaminergic signal transduction within the nigrostriatal pathways are at least partly to blame for schizophrenia, among other things. And those other things also include abnormalities in the glutamate and GABA neurotransmission processes. Scientists have noticed that when the NMDA receptor antagonists are applied to things like ketamine, these can cause psychotic states in otherwise quote-unquote healthy, normal people. The next real-world application of ion channels and neuronal communication is in Parkinson's disease. Parkinson's disease is the second most commonly occurring neurodegenerative disorder. This disorder involves the progressive degeneration of dopaminergic neurons in the brain, meaning that once degeneration starts, it is a progressive ongoing process. It cannot be reversed or cured at this point in time. Some evidence suggests that ion channels may play fairly significant roles in the progression, meaning getting progressively worse, and the pathogenesis of this disease. Parkinson's has 4 cardinal motor losses that are linked with the degeneration of the dopaminergic neurons. These 4 cardinal motor losses are bradykinesia, rigidity, postural instability, and the hallmark symptom, which is typically the tremor. Only 2 of these 4 cardinal motor symptoms must be present in order to receive a Parkinson's diagnosis. In conclusion, ions flow down the concentration gradient. They move from areas of high concentration to areas of low concentration. So when that voltage gated sodium channel opens, it creates a change in the permeability in which the inside of the cell has a lower concentration than the outside of the cell. This creates a concentration and electrically friendly area for the sodium ions to move through. It becomes far easier for the sodium ions to move into the cell than to stay where they are. Now, this movement of ions into the cell creates an action potential. Secondly, sodium ions moves in due to the concentration favorability. Now, when those sodium ions move in, it triggers a neurotransmitter to be released. Remember that neurotransmitters can be things such as dopamine, serotonin, melatonin, among many others. Neurotransmitters are then sent across that synaptic cleft where they bind to a receptor, to that ligand. Oh boy. To that ligand gated ion channels. If you made it this far, thank you. Malfunctions of these ion channels are at least partially responsible for several diseases. And they're not just listed to the 2 diseases that I named in this lecture. I would encourage you to go research other diseases that are due in at least part to the ion flow. These can include things such as multiple sclerosis, which occurs when the myelinated sheaths of neurons become degenerated. Alzheimer's also came up is something that popped up in my research. I believe Huntington's disease popped up as well, but don't quote me on that because that would be a bad idea. Again, in conclusion, remember that ions flow from high to low. Ions are responsible to trigger the neurotransmitters, which then move across that synaptic cleft, bind, and that makes us do everything that needs to be done in our bodies. Thank you so much for listening. Have a wonderful night.